All right. Next up on our schedule is Hubert from the company Daiichi Sankyo, and I will just make one comment before he begins, and that is, it's, as I said before, it's very important to PXE International that the work that we do is very ethical, that we're very thoughtful, that we have first and foremost people in mind, and we are going to be your advocate to the last moment, to the last mile. Hubert came to join us at PXE research meetings quite a few years ago. We really loved his curiosity, his openness, his interest, and his forthcomingness. And so PXE International is working very closely with him now, as is Yoni's group. We're working together on a big project, and we're very happy to have such a great company, and particularly Hubert working with us. Without further ado, Hubert.
Well, thank you very much, Sharon. I really appreciate that introduction. So it's really a privilege to have this opportunity to share with the community here on how we're evaluating a TNAP inhibitor as a potential treatment for PXE. But before I begin, I'll just say that, as a conflict of interest or potential, that I am an employee of Daiichi Sankyo. This is a company, as Sharon said, that is developing novel agents for ectopic calcification, including PXE. And the view is opinion I'm expressing here in this conference of that of my own. But to emphasize, it's really an honor to speak with you all and to have this chat. But before I begin, I would just say a little bit self-introduction. I consider myself a physician scientist. My medical training is in internal medicine and endocrinology. And I've been in the drug development arena for about 20 years. I've been in Daiichi Sankyo for the last 12 years or so. I consider myself a clinical trialist. But when I'm not doing that, this is what I do. I love every now and then beer and wine, of course, and good spicy food, but horseback riding, as you have seen earlier, the background that I have, the mountain of Montana. But when I'm not doing that, I've been thinking about, especially in the last 5-6 years, as Sharon alluded to, about PXE, about TNAP inhibitor, and how that might be useful for PXE treatment. So what exactly is TNAP or TNAP inhibitor? So TNAP really stands for tissue non-specific alkaline phosphatases. And so we're developing an inhibitor to this. And the roles of this TNAP in bone and tissue metabolism, just a few points here. One, the majority of the blood alkaline phosphatase, just like in your laboratory test, you'll see the alkaline phosphatase. Majority of those in circulation comes from TNAP. And TNAP is the enzyme that controls the pyrophosphate level by removing the pyrophosphate. It cleaves it and inactivates it. And so in that, and you have heard a lot about pyrophosphate or PPI that regulates the calcium deposit in bones and limit the abnormal deposit in soft tissue. So pyrophosphate, as you heard from Yoni, from many others, is really central to the story of PXE development. And we know individuals with PXE have low level of pyrophosphate and have abnormal calcium deposit. So that's a lot of words. So it's, schematically, this is what you have seen throughout the last yesterday and today. The ABCC6 as a channel that allows ATP to be released, and that the first enzyme to cleave into pyrophosphate, and also another component, AMP, which is subsequently degraded with the second enzyme. For pyrophosphate, it's degraded through this TNAP. So it stands to reason that if we were to inhibit or reduce the activity of this TNAP, we can increase the pyrophosphate level and hence reduce this calcium deposit. So is that really true? I mean, this is wonderful as on this schematic on the slide, but certainly there are a huge amount of work that has gone into this and show that this scientifically, at least in the animal model, is quite true. So I'll cite a few. There's strong evidence of use of TNAP inhibitor potentially for PXE. These research I cited 3 different excuse me, line of evidence. One is the use of TNAP inhibitor to reduce calcification in this model, PXE mouse model. And that has been done in many labs, particularly that from Dr. Yui to Dr. Lai, and using this prototypical TNAP inhibitor. The work is independently done also in another lab by Ziegler et al. And in Daiichi Sankyo, we also have performed using the various TNAP inhibitor and confirm that this definitely works in this mouse model. Besides using a TNAP inhibitor, you actually can use a genetic engineering method to reduce the TNAP. And that work, and I will show you quickly in the next slides, as done by Dr. Lai and Dr. Yuto's lab as well. And we in Daiichi Sankyo also have replicated independently that results. And as Dr. Pomozzi earlier shown you, you can use the exogenously or give the pyrophosphate directly to the mice in these animal model. You definitely can show the reduction in calcification. That really say and show again and confirm that pyrophosphate is play a very essential role here. So in the next 2 slides, let me just cite some of the previous experiments that the researchers have done. This is citing the Ziegler's paper. And in this experiment, using this prototypical TNAP inhibitor in the PXE model, or so-called ABC6 knockout, and you can give this compound either right after weaning, all right, before the onset of the calcification. And when you give this compound, the TNAP activity is drastically reduced. Great. Calcification is assessed, as Dr. Yuto mentioned earlier, in the whisker, the base of the whisker of the visa. And you can see that calcification is, as expected, reduced as well. You can do this before calcification starts, or you can do this after calcification, after the onset of calcification. And in this case, you can see, and this is given from week 20 to 36, and you can see at the end of that treatment period, you can see the reduction in calcification, not as dramatically as when it's given early on. But one thing for those of you that may be curious, this certainly has not taken the calcification down to much lower levels. So we don't know, the jury is still out whether any of the calcification that's already there can be reversed or not, at least not in this model, in this experiment. So as I said, this is using a chemical to inhibit the TNAP. And Dr. Lai's lab has done using the engineering, genetic engineering. As you know, each of us has 2 copies of every gene. And you knock out one and ask, with 50%, only 1 copy instead of 2, you have 50% of the activity of TNAP. Can you reduce the calcification? And the answer is yes. This is a wild type, meaning it has 2 copies of the wild type for the TNAP, and you see these calcification. When it's only half of the TNAP, and you see much reduced calcification. Quantification is shown on the right side, and again, you can see a significant reduction. So that's great. So what are these TNAP inhibitors? Certainly there are many in the research world, there have been many TNAP inhibitors being used, but these are the newer generation TNAP inhibitor. The SBI 425, as I said, has been used quite extensively in research work. It's a potent TNAP inhibitor from Sanford Burnham, and Dr. Jorge Luis Milan has been really the giant in this field, pushing forward the potential use in many different animal model beyond the PXE model. And so a lot of foundation being laid down. For Daiichi Sankyo, we have a newer inhibitor, sort of improved version, if you would, that more suitable for clinical testing. And it's potent, it's selective for TNAP comparing to other alkaline phosphatases. It's also orally available, meaning you can take it by mouth instead of having to inject. It has improved pharmaceutical properties for clinical testing. And by that, we think through in these kind of evaluation in before human testing, pharmacology, from the test tube to the cell culture to the animal model, those being done on what's called the CMC. Can we really make this compound, and can we make it in a controlled fashion that it's uniform? That has been done. It's a chemical. Does it have the property that you can, when you take it by mouth, can it be absorbed? Can it be, can it show up in the bloodstream as intended? Would it be eliminated quickly? Or would it be metabolized to generate something else that you have to worry about, or at least have to characterize? Those being done. How about how safe is this? So that, what we call the preclinical safety assessment, that has been done as well in at least 2 different animals and taken from a short-term single dose, 1 day, to 3 months, then to longer term. So we have a compound that's selected for clinical testing now. And with that, we have taken that into the healthy volunteers, so-called phase 1 study. And in these kind of so-called phase 1 evaluation in healthy volunteers, typically what's done is what's called single ascending dose. So you give each volunteer just 1 dose. And the schematically shown on the right, and this is a slow, methodical, stepwise process. You give them the lowest possible dose, this designated as 1. And once you've given that with the control, people that receive sugar pill, you test 3 times after you know that the first dose is safe, of the first cohort. And then you go 3 times higher, then you go 10 times higher comparing the first dose, you go further up 30 times, 100 times, until you potentially see something that you have to stop, or as you plan the highest dose. So these are done, and this up to the single dose, you give each of these cohort multiple doses, and it could be repeated daily up to, for 7 days, for 10 days, or 2 weeks. You're gonna have to test the formulation that you want, the tablet formulation you want, for example. And these are all done randomized, double-blind, and placebo-controlled. That's really to take away the potential bias, either from the participant, from the personnel administrating the medication, or from the sponsor for that matter, outside of their safety assessment. So with that, we assess really the safety tolerability. We understand what's called the pharmacokinetics, meaning the drug gets in and gets out, and we confirm the intended mechanism. Does it really work? And what we do know for this evaluation in phase 1 that we have completed, that the TNAP activity can be inhibited as schematically shown here, and concomitant with that, the pyrophosphate level can increase. So this is a really great confirmation. And that what we learned from this phase 1 evaluation is that TNAP inhibitor that we're using is safe, at least in these short-term studies. And we have dose in a single dose, more than 100 times higher than the likely dose that we will be taking forward. In the multiple dose setting, we have taken more than 10 times higher. So that provides a safety assurance for the amount that we know will be safe, even if it's much higher. We know that the expected presence is there for the drug, and it's consistent with the mechanism of action of TNAP inhibition. So we're taking this forward. We think to support the further development. So what does the typical phase 2 look like then? So Again, it's usually randomized, placebo-controlled, double-blind. And in the typical setting, maybe placebo plus 2 different doses, dose A and B. The safety is always most important. The PK, the pharmacodynamics or the biomarkers, and the clinical responses. In this case, it may be taken for a total of weeks. So a whole bunch of assessment that will be done, exam, routine test, the drug concentration, and other biomarker. So when someone is eligible to participate, they will be randomly assigned into these 3 different groups and come in for different clinic visits for the exam, right? So beyond the phase 1, it's very short term in healthy volunteer. In phase 2, a little bit longer term in the population that you want to study, in this case, PXE. There also need to be taken then longer term into phase 3. And here, and that will be a much longer evaluation using the various different clinical endpoints, whether that's calcium in the CT scan, Dr. Wilco Spearing has mentioned, or other assessment. But I'll make a pitch, as you heard from Sharon earlier, that many of you have participated in the registry study, really helping to understand what kind of signs, symptoms, timing of the signs and symptoms that you might have. There's an effort really to organize beyond the data collected in your routine clinical care, a more regimented, more prospective, and carefully collected and uniform collection of data. And that's what we call the PXE natural history study. And I'm sure you'll be hearing more as this being organized. So coming back to this, I think now this is really an exciting time, and what I have taken you through is an example of the development of the potential therapy and evaluation of this therapy and process. And we're focusing on this TNAP inhibitor. But certainly there are many other targets that you have heard, whether that's directly working on the ABCC6 itself, or working on the enzyme that produces the pyrophosphate, or the pyrophosphate itself with either taken by mouth or other analog of pyrophosphate. And lastly, as Dr. Yuito mentioned earlier, can we do something about this calcium deposit? So there are many targets that the whole community is working on, and this is really an exciting time. But we're not quite there yet. This process is going to take a long time. This is not easy. And just take a look at, while it's exciting, it's still very challenging. What are the roadblocks, and what are the data that show us that we still need still have a journey to go, right? And I like to show you this historic data on what's called the probability of success in drug development. And this is data, US data. And looking at this, the way it's constructed is different drugs development in different therapeutic areas, right? And maybe the musculoskeleton is somewhere in the middle, but for the compound enter the phase 2 development, the success rate for approval, it's only about 10 to 20%. And if it's for those that enter phase 1, even lower, less than 10%. And hopefully, and these certainly are driven, every drug is different. But even those that enter phase 3, we think it's really good coming from phase 1 and phase 2. There are some assurances of the safety, at least in those duration of short term in phase 2 study. It's still a coin toss to some degree, about 50%. So it's, there are many things still need to be done. But I think, even with that, this is a very exciting time because, as I mentioned, there are so many different mechanisms that it's a race from everyone, from the entire research community, trying to develop a potential therapy. And that's built on really 25 plus years of research in this field that the entire community have done. And I would say in the coming decade, will be really exciting as many of these things are entering into the clinical phase, and how we capture and assess how these potential therapy can or cannot work, whether it's short-term using pyrophosphate or using the calcium deposit by CT scan. At the end of the day, we still need to show that these potential therapy will really improve how you feel as a PXE, how you function, and that's really the most important part, and so that we can decrease the disease burden itself. And I just make a pitch that it really takes a huge global village from people participating together, from the PXE, the family, the patient advocacy group, the PXE International, the basic science researchers, the clinical science researcher, the academia, and even biotech and pharmaceutical company. It really takes a global village effort to get this done. And in that, I think a lot of things really could not have happened without the leadership of the PXE International and Sharon. So thank you. Thank you again for the opportunity to share with you the TNAP, the evaluation of the TNAP inhibitor.
Great, thanks very much. Hubert, always good to get that kind of description. I'm really happy that you went through those steps, because those are going to be very critical, very important to us as we do move various potential therapies into the treatment arena. And you can see there were a lot of steps. You see there's a lot of safety built into this. You've been hearing a lot about this lately because the vaccine development that's in the news in a major way is going through all these same sorts of steps. And when you hear the trial has been stopped, the Data Safety Monitoring Board has found one sick person out of 30,000. You can see how careful these drugs are in their development, and something like these vaccines really get a ton of scrutiny. And the same thing's happening here, even in our small picture. And again, we don't have 30,000 people to participate in a PXE trial. We're going to need every single one of you. Any questions? We're running a little over, but we can take, looks good. Okay. All right. Thanks so much, Hubert.
Thank you.